380 million years ago, nothing on the surface of this planet would have been recognizable. The continents sat in configurations that no longer exist. The atmosphere carried oxygen at concentrations measurably lower than today. The land, for the most part, was still barren. Low, primitive vegetation clinging to shorelines, vast interiors of exposed rock and sediment baking under an unfiltered sun. There were no forests yet, no large animals moving across open ground, no sound from the land beyond wind and water. But beneath the surface of the ancient seas, something else entirely was taking shape. The oceans of the Devonian period were not empty. They were not quiet. They were some of the most biologically dense, ecologically complex, and lethally dangerous environments this planet has ever produced. And at the center of those ecosystems, at the absolute top of the food chain, moved an animal that has no true equivalent in the world today. It was armored, it was massive, and its jaws could generate forces capable of crushing through bone, shell, and solid plate in a fraction of a second. This is the world of Dunkleosteus, and it was already ancient by the time the first trees began to grow. The Devonian period spans from approximately 419 million years ago to 359 million years before the present. A duration of roughly 60 million years during which the biological character of this planet was transformed at nearly every level. In the scientific literature, this interval is frequently designated the Age of Fishes, a label that reflects the extraordinary diversification of fish lineages across the period. But that designation, accurate as it is, does not fully capture what the Devonian oceans actually were. Fish diversification was not occurring in a vacuum. It was occurring within ecosystems of rapidly increasing complexity under predation pressure of escalating intensity in environments shaped by reef structures, sediment, dynamics, temperature gradients, and biological competition operating simultaneously across hundreds of interacting species. The shallow seas of the Devonian were warm. Surface water temperatures in tropical and subtropical zones are estimated to have ranged between 25 and 35 degrees Celsius across much of the period. In open water, sunlight penetrated the upper column with relative clarity, but in the zones where biological activity was most concentrated, near reef structures, river discharge points, and coastal shelves, suspended sediment, algal material, and organic particulate reduced visibility substantially. These were not clean, open waters. They were layered, structurally complex environments in which predator and prey operated within constrained detection distances. That constraint shaped everything about the way predation worked in Devonian seas. It is within this context that the placoderms must be understood. Placoderms were a class of jawed fish defined by a rigid bony shield covering the head and the forward portion of the trunk, composed of interlocking dermal bone plates. The rear section of the body remained flexible, either lightly scaled or unarmored. The critical anatomical feature was a functional hinge between the head shield and the trunk shield, a joint that allowed the entire cranium to rotate upward as the lower jaw dropped. This mechanism dramatically increased the volume of the oral cavity during prey capture and contributed to the generation of powerful suction. It was an evolutionary solution to the problem of securing fast-moving prey in open water, and it worked with measurable efficiency. Among all placoderms, one genus became the defining predator of the late Devonian seas. Dunkleosteus terrelli, first described from fossil material recovered in late Devonian deposits of what is now Ohio, is the largest and most extensively studied species within its genus. Size estimates have been complicated by a persistent problem in the fossil record. The heavily mineralized head and trunk shields preserve reliably, but the posterior sections of the body, less mineralized, more prone to decay, rarely survive intact. Complete body fossils have not been recovered. Current estimates, based on proportional comparisons with better preserved placoderm relatives and scaling relationships with modern analogues, place large individuals of Dunkleosteus terrelli at between 4 and 6 meters in total body length, with some analyses indicating the possibility of individuals approaching 8 meters. Mass estimates remain uncertain but are consistent with an animal of substantial physical presence and significant inertial momentum in water. The oral anatomy of Dunkleosteus was unlike that of any fish currently alive, rather than true teeth. The discrete, individually rooted mineralized structures found in sharks and modern bony fish. Dunkleosteus possessed self-sharpening cutting blades formed directly from the edges of the jaw bones themselves. These blades were composed of bone, and their geometry was maintained through wear use, where during feeding preserved a consistently sharp cutting edge, through a mechanical principle comparable to that observed in the self-sharpening incisors of certain rodent lineages. The anterior portion of the jaw carried fang-like projections capable of piercing and securing prey. The posterior surfaces provided a shearing action capable of processing heavily armored or hard-shelled prey items that most contemporaneous predators could not manage. Biomechanical modeling of the jaw musculature and lever geometry based on preserved skull material has produced bite force estimates ranging from approximately 4,000 to over 7,000 newtons at the anterior jaw tips, with considerably higher forces calculated at the posterior shearing surfaces. These figures place Dunkleosteus among the most forceful biters documented in Devonian marine environments. The speed of jaw actuation has been estimated from the mechanics of the cranial hinge system. Full gape could be achieved in approximately 50 to 60 milliseconds under maximum muscle activation, producing a rapid pressure drop at the mouth opening that drew water and prey inward before an evasive response could be initiated. The evidence for what Dunkleosteus actually consumed comes from multiple fossil sources. Partially digested fish remains, including identifiable placoderm fragments, have been recovered in association with specimens in positions consistent with stomach contents. Regurgitated boluses. Compacted masses of indigestible hard parts expelled from the stomach rather than processed through the full digestive tract have been recovered from late Devonian deposits alongside Dunkleosteus material. These boluses contain the crushed remains of armored fish whose own defensive structures did not prevent predation. The feeding range implied by this evidence is broad. Dunkleosteus was not a specialist on soft-bodied prey. It was processing some of the most heavily defended animals in its environment. The reef systems within which Dunkleosteus hunted were built by organisms entirely unlike those constructing modern reefs. The dominant reef builders of the Devonian were stromatoporoids, sponge-grade organisms with layered calcareous skeletons. Combined with tabulate and rugose corals, both groups now completely extinct. These structures created three-dimensional habitat complexity across shallow tropical and subtropical seafloors, concentrating biological activity and establishing zones of high prey density within architecturally complex environments. Brachiopods, crinoids, trilobites, mollusks, and dozens of fish species occupied these reef systems in overlapping ecological roles, forming communities in which predation pressure was high and spatially structured by the geometry of the reef itself. In low visibility conditions near reef margins, where sediment input from river systems draining increasingly vegetated Devonian landscapes reduced detection distances. A predator of Dunkleosteus's body mass and jaw speed had a measurable advantage. Prey reaction times are constrained by the distance at which a threat can be detected. Reduce that distance, and the margin available for evasion narrows accordingly. The mechanics of Dunkleosteus's strike. Rapid gape expansion, high suction force, anterior fang engagement within the first fraction of a second were well matched to the conditions of its environment. Beyond the reef margins, conditions changed. Light availability decreased with depth. Oxygen concentrations in some deeper basins were reduced. The biological community shifted toward organisms tolerant of lower productivity and reduced visibility. Large-bodied predators like Dunkleosteus likely traversed the boundary between shallow reef environments and deeper water, exploiting prey concentrations in productive zones while operating across the full range of the shelf environment. Alongside the placoderms, the Devonian seas supported early representatives of nearly every major fish lineage that would persist into subsequent periods. Members of the class chondrichthyes, defined by cartilaginous rather than bony skeletons, were present throughout the Devonian, though their ecological dominance during this period was substantially less than it would become after the end Devonian extinctions reshaped the competitive landscape. Devonian shark taxa were, in general, smaller than the apex placoderms, and the available evidence places them in somewhat different ecological roles. Processing invertebrates or functioning as mid-level predators, rather than in direct competition with Dunkleosteus-grade animals for the same prey at the same scale. The coexistence of ecologically diverse placoderm forms, early sharks, and a wide range of non-placoderm fish lineages within the same Devonian marine environments reflects the degree of niche differentiation that had developed across tens of millions of years of evolutionary diversification. Fossil assemblages from key late Devonian localities. Sites in Morocco, Poland, Ohio, and eastern Canada preserve the remains of dozens of fish species alongside equally diverse invertebrate faunas, recording snapshots of ecosystems in which competition and predation were operating with high intensity across every ecological level simultaneously. The late Devonian seas, in the interval between approximately 385 and 359 million years ago, were environments of extraordinary biological density. The selective pressure they imposed toward more effective predation, more effective defense, faster responses, better sensory systems was continuous and intense. Every structural feature of Dunkleosteus, from the geometry of its jaw blades to the speed of its cranial hinge, reflected millions of years of refinement within exactly this kind of environment. These were not primitive ecosystems in any sense that implies simplicity. The product of biological processes that had been running without interruption for hundreds of millions of years before the first armored predator broke the surface of a Devonian sea. Step back from the ocean's edge. Turn away from the reef systems, the armored predators, the layered biological complexity of Devonian seas. Look instead toward the land, toward the vast continental interiors that stretched away from the coastlines in every direction, largely untouched by the biological transformation occurring in the water. What existed there, for most of the Devonian, was close to nothing. Not absolute nothing. The land was not sterile. Microbial mats had colonized damp surfaces near water for hundreds of millions of years before the Devonian began. Primitive, non-vascular plants. Organisms broadly comparable to modern liverworts and mosses had established a thin biological presence along shorelines and riverbanks during the preceding Silurian period, and that presence persisted into the early Devonian. But in any meaningful ecological sense, the continental interiors of early Devonian Earth were empty. There were no trees, no shrubs, no ground cover of any structural complexity, no large animals moving across open terrain. The land surface, across most of its extent, was exposed rock and sediment, fractured, weathered, and subject to erosion at rates that modern landscapes, stabilized by deep root systems and continuous vegetative cover, do not experience. This was not a transitional moment between two forested worlds. It was a fundamentally different planetary condition, one that had persisted with only marginal change for the vast majority of Earth's history to that point. The physical consequences of a largely unvegetated land surface were significant and measurable. Without root systems penetrating and binding sediment, erosion operated with a directness and efficiency that shaped the character of Devonian river systems, coastlines, and shallow marine environments in ways directly visible in the sedimentary record. Devonian river deposits, preserved in sequences across North America, Europe, and elsewhere, frequently exhibit the broad, shallow, braided channel geometries characteristic of systems carrying high sediment loads across low-cohesion floodplains. Modern braided rivers of this type are typically found in high sediment environments. Glacial outwash plains, recently deglaciated terrains, volcanic landscapes, where vegetation has not yet stabilized the substrate. In the early Devonian, this was the default condition across most continental surfaces. Sediment discharged from these river systems into coastal marine environments contributed to the reduced visibility conditions that characterized many Devonian nearshore zones. The connection between the barren continental interior and the ecology of coastal seas was, therefore, direct. The absence of vegetation on land was a physical input into the marine ecosystem, shaping the optical and chemical conditions of the water in which Devonian marine organisms lived and died. The chemical environment of Devonian soils, to the extent that true soils existed, was also distinct from modern conditions. Soil formation, in the modern sense, depends substantially on the decomposition of organic material by fungi, bacteria, and invertebrates living within a matrix of mineral particles and root-derived structure. Early Devonian substrates lacked the biological inputs necessary to produce soils of this kind at any significant depth or geographic extent. What existed were thin, poorly developed weathering profiles. Mineral surfaces partially altered by chemical interaction with water and atmospheric gases, but lacking the organic horizon that defines modern productive soils. The capacity of these substrates to retain moisture, resist erosion, and support plant colonization was correspondingly limited. The plants that did exist on early Devonian land were small, structurally simple, and confined to environments where water was consistently available. Organisms possessing internal conducting tissue capable of transporting water and dissolved nutrients appear in the fossil record from the late Silurian and proliferate through the early Devonian. These plants, belonging to groups such as the rhiniophytes and related lineages, known from exceptional preservation at sites including the Rhynie Chert in Scotland, were typically only a few centimeters to perhaps 20 or 30 centimeters in height. They lacked true leaves in the botanical sense, the broad, flat photosynthetic surfaces that characterize the majority of modern plant lineages. Instead, they possessed simple branching axes, sometimes bearing terminal sporangia for reproduction, photosynthesizing across the surface of their stems. Root systems, in the strict anatomical sense, were absent or rudimentary. These plants anchored themselves through structures called rhizoids, or simple rhizomes that penetrated the substrate to limited depth. The ecological footprint of these early vascular plants was narrow. They could colonize damp, low-competition surfaces near water, riverbanks, lakeshores, the margins of coastal wetlands. But they had no mechanism for establishing themselves across dry or seasonally arid terrain. The continental interiors, away from reliable surface water, remained beyond their reach. The result was a landscape organized around water availability in a more absolute way than any modern terrestrial environment. Near permanent water, a thin fringe of low vegetation. Beyond that fringe, bare substrate extending to the horizon. Sunlight fell on these surfaces without the attenuation that forest canopies produce in modern environments. Surface temperatures in exposed areas would have fluctuated more widely day and night and between seasons than in vegetated landscapes where plant transpiration moderates local humidity and canopy cover reduces direct solar heating of the ground. The continental interiors of early Devonian Earth were, in thermal terms, more extreme environments than comparably positioned landscapes today. Not necessarily hotter in an absolute sense, but more variable, less buffered, and less hospitable to any organism requiring stable moisture conditions. The atmosphere above these landscapes was itself in a state of measurable transition. Oxygen concentrations during the early Devonian are estimated to have been lower than modern levels. Current models suggest values in the range of 15 to 16% by volume, compared to the approximately 21% that characterizes the modern atmosphere. Carbon dioxide concentrations were substantially higher, though estimates vary depending on the proxy methods employed and the specific interval within the Devonian being assessed. These atmospheric conditions had direct biological implications. Lower oxygen, partial pressure, reduced the energetic efficiency of aerobic respiration for any organism operating in air rather than water, a relevant constraint for the earliest arthropods and primitive plant tissues exposed to the atmosphere. Higher carbon dioxide concentrations, combined with increasing plant colonization of land surfaces, were setting the conditions for a series of geochemical feedbacks that would, over the course of the Devonian, alter both the atmosphere and the global climate in ways that no organism alive at the time had any capacity to anticipate or respond to except through the slow mechanism of evolutionary change. The ultraviolet radiation environment at the land surface was also distinct from modern conditions. The ozone layer generated by photochemical reactions involving atmospheric oxygen provides the primary shield against high-energy ultraviolet radiation reaching Earth's surface. At lower atmospheric oxygen concentrations, ozone production rates are reduced and the degree of ultraviolet shielding is correspondingly less complete. Early Devonian land surfaces may have experienced higher ultraviolet flux than modern environments at equivalent latitudes, a physical condition that would have imposed additional stress on organisms attempting to colonize exposed terrestrial substrates without the protection of water or dense vegetative cover. What is most significant about the early Devonian land surface, considered as a whole, is not any single physical characteristic, but the combination of conditions it represented: high erosion rates, poorly developed soils, limited and spatially constrained vegetation, extreme surface temperature variability, elevated ultraviolet exposure, and a lower oxygen atmosphere together produced a terrestrial environment in which the barriers to colonization by complex multicellular life were substantial and overlapping. Any organism capable of surviving on land had to contend not with one unfavorable condition but with several simultaneously, and had to do so using biological machinery that had, in almost every case, been refined over millions of years for function in water. The oceans had been the crucible of complex life for hundreds of millions of years. They moderated temperature. They provided buoyancy. They filtered radiation. They carried dissolved oxygen to respiratory surfaces through passive diffusion and active ventilation. The land offered none of these services automatically. Every function that water performed passively for aquatic organisms had to be replaced, on land, by biological structures that did not yet exist in any fully functional form. That transformation, the gradual, uneven, biologically costly process by which life began to make the land habitable, was already beginning, in the earliest and most tentative way, along the waterlogged margins of Devonian continents. It would take tens of millions of years to complete. And it would change everything. The Devonian seas were not dominated by a single predator operating in isolation. Dunkleosteus was the largest, the most heavily armed, and by most measures the most formidable vertebrate predator in its environment. But the oceans that surrounded it were populated by an extraordinary range of organisms. Many of them dangerous, many of them adapted to exploit the same productive coastal and reef-associated zones, and many of them capable of making survival difficult for animals far smaller and less defended than themselves. To understand the ecology of Devonian seas is to understand that danger was not concentrated at the apex of the food chain. It was distributed across nearly every level of it. The shallow marine environments of the Devonian, the reef platforms, the coastal shelves, the estuarine transition zones where fresh water systems discharged into open sea, were among the most biologically productive environments on the planet during this period. Productivity, in ecological terms, means energy, and concentrated energy in any ecosystem means concentrated competition. Every organism present in these environments was under continuous pressure from predators, competitors, and the physical constraints of the environment itself. The result was a set of ecosystems shaped, at every level, by the sustained intensity of that pressure. The arthropods of the Devonian seas represent one of the most significant and frequently underestimated components of the period's marine fauna. Sea scorpions, Eurypterids, had been present in marine and brackish environments since the Ordovician, and by the Devonian several lineages had reached body sizes that placed them among the largest arthropods in the history of life on Earth. The genus Pterygotus, documented from Devonian deposits across multiple paleocontinents, reached estimated body lengths of approximately 1.5 meters. Jacalopterus rhenanii, known from slightly earlier Devonian-adjacent deposits, has been estimated at up to 2.5 meters based on fragmentary fossil material, a figure that, if accurate, would make it the largest arthropod yet documented in the fossil record. These were not passive filter feeders or slow-moving bottom dwellers. Eurypterids possessed large compound eyes, articulated chelicerae capable of seizing and processing prey, and a body plan that, in the larger predatory forms, was structured for active pursuit in shallow water. The ecological role of large Eurypterids in Devonian coastal and estuarine environments was likely that of generalist ambush or pursuit predators targeting fish, smaller arthropods, and soft-bodied invertebrates in the shallow, structurally complex zones they preferred. Their tolerance for reduced salinity, evidenced by fossil occurrences in deposits interpreted as brackish or freshwater-influenced, placed them in exactly the transitional environments where fish moving between marine and freshwater systems would have been most concentrated and most vulnerable. The decline of large Eurypterids across the Devonian is generally attributed, at least in part, to competitive displacement by diversifying fish lineages, particularly the jawed fish, whose expanding ecological roles overlapped increasingly with those of large predatory arthropods. This transition was not instantaneous. 4. A substantial portion of the Devonian, Eurypterids and placoderms coexisted in shared coastal environments, and the interactions between these groups would have constituted a significant component of the predation landscape experienced by smaller organisms in those zones. The sharks of the Devonian require separate consideration because they're Devonian representative from the forms that would come to define the group in later geological periods. Early Devonian shark lineages, including forms such as Doliodus problematicus, known from exceptionally preserved material from New Brunswick, Canada, and currently regarded as among the oldest well-documented shark specimens, possessed both pectoral fin spines and a dentition suggesting a generalist predatory role. By the Middle and Late Devonian, shark diversity had expanded considerably, with taxa exhibiting a range of body forms and dental morphologies that imply ecological differentiation across feeding strategies. Ctenacanthiform sharks, present in late Devonian deposits, were relatively robust-bodied forms with crushing dentition in some lineages, suggesting a capacity for processing hard-shelled prey. Other lineages possessed more blade-like teeth, consistent with a diet of soft-bodied fish or invertebrates. What is notable about Devonian sharks is not their individual size. Most were considerably smaller than the large placoderms they coexisted with, but their sensory and hydrodynamic capabilities. The cartilaginous skeleton of chondrichthyans, less energetically costly to produce than bone, combined with a body plan refined for efficient locomotion through water, gave early sharks physical characteristics well suited to active pursuit predation in open water. The lateral line system and electroreceptive ampullae present in shark lineages provided sensory information about water movement and bioelectric fields that extended effective detection range beyond what vision alone could provide in low-clarity water. In the murky coastal and estuarine environments of the Devonian, these sensory capabilities represented a measurable ecological advantage. The coexistence of early sharks, large Eurypterids, and apex placoderms in Devonian coastal environments created a predation landscape of considerable complexity. Smaller fish navigating these zones were not contending with a single type of threat from a predictable direction. They were operating within environments in which threats varied in size, sensory capability, attack strategy, and preferred habitat zone. A combination of pressures that drove the diversification of defensive adaptations across prey lineages with corresponding intensity. The reef environments of the Devonian added further complexity to this already dangerous ecosystem. Devonian reef structures, built primarily by stromatoporoids and extinct coral lineages, were among the most extensive reef systems in Earth's history. During the Middle Devonian in particular, reef development reached a global scale, with barrier and platform reef systems extending across shallow tropical seas in what are now North America, Europe, and Australia. These structures were not simply physical habitat, concentrating filter feeders, grazers, and the predators that fed on them into spatially defined zones of high ecological activity. Within reef environments, visibility conditions varied dramatically over short distances. Open water above the reef crest received relatively unobstructed sunlight and offered the greatest detection distances. Among overhangs, in channels between coral and stromatoporoid masses along the rubble zones at reef bases, light was reduced, current patterns were complex, and the spatial geometry created numerous concealment opportunities. Predators adapted to exploit these conditions did not need to pursue prey over long distances. They needed only to position themselves appropriately and wait. The fossil record of reef-associated Devonian environments reflects the biological density of these systems. Locality, such as the Frasnian deposits of the Canning Basin in Western Australia, and the reef-associated sequences of the Eifel region in Germany have yielded faunal assemblages of extraordinary richness. Preserving the remains of dozens of fish species, abundant invertebrate fauna, and the skeletal remains of the reef builders themselves in near-original spatial relationships. Dozens of these assemblages document ecosystems in which the physical structure of the reef was inseparable from the ecological dynamics playing out within it. Away from the reefs, in the open shelf environments and deeper basinal waters, different pressures applied. In lower oxygen bottom waters, large predators were less active, but the organisms that tolerated these conditions, certain trilobite lineages, deposit-feeding invertebrates, and some fish taxa, were themselves subject to predation from animals capable of descending into marginally oxygenated zones. The boundary between well-oxygenated shelf waters and the oxygen-depleted bottom environments of some Devonian basins was a sharp chemical gradient that influenced the distribution of nearly every organism in the water column above it. Storms were a regular feature of Devonian coastal environments. Epicontinental seas, lacking the depth that buffers modern ocean basins from atmospheric disturbance, were subject to wave action and sediment resuspension during storm events that could temporarily transform visibility conditions, displace organisms from preferred habitat zones, and concentrate prey encounter rates in ways that altered predator-prey encounter rates. Evidence for storm influence is preserved in Devonian sedimentary sequences as graded beds, hummocky cross-stratification, and event layers of coarser material module interrupting finer background sedimentation. These physical records indicate that environmental instability was not an occasional disruption but a recurring condition that organisms in Devonian shallow marine environments had to persist through repeatedly. The cumulative effect of these overlapping pressures, apex vertebrate predators, large arthropod competitors, early sharks with refined sensory systems, structurally complex reef environments offering concealment to ambush predators, and recurring physical disturbance from storms and sediment events, was a marine ecosystem in which survival was never a condition of stable equilibrium. It was an outcome continuously negotiated against a background of active multi-directional threat. Every adaptation visible in the Devonian fossil record, armor plated, increased jaw complexity, enhanced sensory systems, behavioral specialization toward particular habitat zones, reflects the selective force of these pressures acting across millions of years of biological time. The Devonian seas were not simply dangerous because of the size of their predators. They were dangerous because danger had been built into the structure of the ecosystem itself, layer by layer, over tens of millions of years, until the ocean was less a habitat than a continuously operating system of elimination, in which only the most precisely adapted survived long enough to leave a record in stone. The transition from water to land was not an event. It was not a moment of biological decision, a sudden crossing from one world into another. It was a process, incremental, uneven, and extended across millions of years, in which the boundary between aquatic and terrestrial existence was crossed not once but repeatedly by different lineages, in different ways, under different environmental pressures. The fossil record does not preserve a single first step. It preserves a gradual blurring of the line between fish and something else. That blurring began in the water. By the Middle and Late Devonian, certain lineages of lobe-finned fish, members of the group Sarcopterygi, characterized by fleshy, muscular fins supported by an internal bone structure homologous to the limb skeleton of land vertebrates, had developed anatomical features that extended their functional range beyond open water. The key lineages in this transition are documented primarily from fossil material recovered in late Devonian deposits across eastern North America, Greenland, and Latvia, and they include some of the most consequential specimens in the vertebrate fossil record. Tiktaalik rosier, described from Upper Devonian deposits of Ellesmere Island in the Canadian Arctic and estimated at between 1 and 2.75 meters in body length, occupied a morphological and ecological position directly relevant to the water-to-land transition. Tiktaalik possessed gills and scales consistent with an aquatic lifestyle, but also lungs capable of aerial respiration and a pectoral fin skeleton that included a functional wrist-like joint, allowing the fin to be used for propping and limited weight-bearing. The neck. The neck, absent as a distinct anatomical region in most fish, whose head and shoulder girdle are rigidly connected, was present in Tiktaalik as a genuinely mobile structure, allowing independent movement of the head relative to the body. This feature has direct functional relevance to feeding and navigation in shallow, structurally complex environments where maneuverability matters more than sustained open water speed. The environment in which Tiktaalik and related forms lived was not open ocean. Evidence from the sedimentary context of Tiktaalik fossils indicates deposition in a subtropical, seasonally variable floodplain environment. Shallow streams, mudflats, and vegetated margins where water depth was frequently insufficient for sustained swimming, and where the ability to prop the body above the substrate, navigate around obstacles, and access air directly would have provided measurable advantages. These were environments at the interface of water and land, and the organisms that inhabited them were under selection pressure from both directions simultaneously. Approximately contemporaneous with Tiktaalik and known from late Devonian deposits in Latvia, the genus Ventastega represents as another point along the morphological continuum between lobe-finned fish and fully limbed tetrapods. Ventastega possessed a skull and pectoral girdle anatomy intermediate between the fish-grade condition and that of the earliest true tetrapods, and is interpreted as occupying a broadly similar ecological zone. Shallow, vegetated, freshwater or brackish environments in which the physical demands of navigating complex substrate drove anatomical change over successive generations. The earliest confirmed tetrapods, animals with 4 discrete limbs bearing defined digits rather than fin rays, appear in the late Devonian fossil record from multiple geographic locations within a relatively narrow stratigraphic interval. Ichthyostega and Acanthostega, both known from Upper Devonian deposits in Greenland, are among the best studied of these early tetrapods, and both have yielded anatomical information that substantially complicates any simple narrative of land conquest. Acanthostega, despite possessing 4 limbs with 8 digits each, shows skeletal features of the limbs and shoulder girdle that are inconsistent with sustained weight-bearing on land. The animal's limbs appear to have been functional primarily in water, used for maneuvering through dense aquatic vegetation, navigating substrate, or generating thrust in shallow environments, rather than for terrestrial locomotion in any extended sense. Ichthyostega, with a more robust skeletal construction and evidence of a different limb posture, may have been capable of limited terrestrial movement, but the details of its locomotion remain a subject of active research and ongoing revision. What these early tetrapods collectively indicate is that limbs evolved within an aquatic context for aquatic functional demands, before they were co-opted for terrestrial locomotion. The land was not the target of this evolutionary process. It was a consequence of it. An environment that became accessible as a byproduct of anatomical changes driven initially by selection in shallow water. The environments along the margins of Devonian water bodies were not welcoming in any straightforward sense. Mudflats exposed at low water, tidal influence zones, and the shallow margins of freshwater systems were physically demanding environments for any organism venturing onto them. Substrate cohesion varied, firm enough in some areas to support body weight, soft enough in others to impede movement significantly. Desiccation was an immediate threat for any organism whose skin provided insufficient barrier to water loss. Temperature at the surface of exposed mudflats under direct Devonian sunlight with minimal atmospheric filtering and no vegetative shade would have fluctuated significantly across the daily cycle, potentially reaching values that imposed thermal stress on organisms with limited thermoregulatory capacity. The available food resources on land were limited, but not entirely absent. Primitive plant material along water margins, arthropods that had independently colonized terrestrial environments in preceding geological periods. Certain myriapod and arachnid lineages are documented from Silurian and early Devonian terrestrial deposits. And organic detritus accumulating at the water's edge all represent potential sources for an organism capable of accessing them. The predation pressure that was intense and omnidirectional in open water was, at least initially, reduced on land. No large vertebrate predator yet operated in terrestrial environments. An animal capable of moving onto exposed substrate, even temporarily, was moving into a space where the competitive and predatory pressures it had evolved to avoid were temporarily absent. This reduction in predation pressure is frequently cited in discussions of the selective context for terrestrial locomotion. And while it cannot be isolated as the sole driver, it represents a plausible component of the ecological incentive structure that made excursions onto land selectively neutral or advantageous even before the anatomical machinery for sustained terrestrial life was fully developed. The fossil trackway record adds a further dimension to this picture. Fossilized trackways interpreted as tetrapod in origin have been recovered from late Devonian and possibly earliest Flaminian deposits in Poland. The Zahelmi trackways, described from the Holy Cross Mountains. These trackways predate by a measurable stratigraphic interval some of the body fossil evidence for early tetrapods, and their morphology indicates an animal moving on 4 discrete limbs across a soft substrate in a shallow coastal or lagoonal environment. The trackways do not preserve the animals that made them, but their existence confirms that limbed vertebrates were moving across soft substrates and leaving physical impressions in sediment by at least the Middle Devonian, earlier than the body fossil record alone would indicate. The water-to-land transition, viewed across its full temporal and biological extent, was among the most consequential processes in the history of vertebrate life. It did not produce, in the Devonian, anything resembling a fully terrestrial animal in the modern sense. What it produced was a set of organisms operating at the margin, biologically committed to water in most functional respects, but increasingly capable of extending their activity into the thin, difficult, resource-limited zone where water ended and land began. Everything that would eventually walk, run, climb, or fly across the surface of this planet descended from animals navigating that margin. They did it slowly, imperfectly, and without direction, but they did it. The land did not become forested all at once. It happened gradually, unevenly, and through a sequence of biological innovations whose cumulative effect was so profound that it altered not just the appearance of the continents but the chemistry of the atmosphere, the structure of the soil, the hydrology of river systems, and the composition of the oceans themselves. The rise of the first forests was not simply a botanical event. It was a planetary transformation, one of the most consequential reorganizations of Earth's surface systems in the last 500 million years. It began, as most large transitions do, with something small. The earliest vascular plants of the Silurian and early Devonian were, as described, structurally simple, low-growing, rootless or near-rootless, confined to waterlogged margins. But across the Middle and Late Devonian, plant lineages diversified with a speed and structural complexity that the preceding terrestrial record had not anticipated. The innovations that drove this diversification were anatomical, and they were interdependent. Each one expanded the range of environments accessible to plants, and that expansion in turn created new selective pressures that drove further innovation. The development of true roots, branching structures capable of anchoring plants in substrate, extracting water and dissolved minerals from soil, and penetrating to depths unreachable by rhizoids, was among the most significant of these innovations. Roots did not simply provide structural support. They actively modified the substrate they inhabited, apart mineral particles through physical expansion and chemical exudate, creating the conditions for soil formation that had not previously existed at any significant scale. As root systems penetrated deeper and spread wider, they produced a biologically active substrate layer that retained moisture, accumulated organic material, and progressively transformed bare mineral sediment into something functionally different. Something approaching soil in the modern sense. Simultaneously, plant lineages were developing the capacity for secondary growth, the production of woody tissue through repeated cell division in a lateral meristem, a process that allowed individual plants to increase in girth over successive growing seasons and thereby support increasing height. Lignin, the complex organic polymer that provides structural rigidity to woody tissue, was a biochemical innovation of enormous ecological consequence. It allowed plants to grow tall enough to access sunlight above competitors, extending upward into a dimension of the environment, vertical space, that had not previously been a competitive arena for terrestrial life. It was also, as would become clear only over geological timescales, extraordinarily resistant to decomposition. By the late Devonian, these innovations had combined to produce the first genuinely arboreal ecosystems. Archaeopteryx, known from fossil material across multiple continents and considered the first tree in the modern ecological sense, reached heights estimated at 8 to 10 meters or more, with a woody trunk produced by secondary growth, a spreading branching architecture, and large, flat, photosynthetically efficient leaves. Archaeopteryx forests are documented from late Devonian deposits in North America, Europe, and elsewhere, and their geographic extent indicates that forested environments had expanded from localized stands along waterways as art to something approaching regional-scale woodland across suitable terrain by the Furmenian stage has. Alongside Archaeopteryx, other plant lineages contributed to the structural diversity of late Devonian vegetation. Lycopsids, the group that includes modern club mosses, produced tree-like forms during the Devonian that reached several meters in height. Progymnosperms, the broader group to which Archaeopteryx belongs, varied considerably in their architecture. Doxolopsids, an extinct group known primarily from Devonian deposits, produced some of the earliest genuinely large plants, with trunk structures unlike anything in the modern flora. Hollow, with complex internal vascular architecture, radiating outward from a central cavity in patterns that have no direct modern analog. The physical environment beneath these early forest canopies was distinct from both the open terrain that had preceded them and the mature forest floors of later geological periods. Light levels beneath a closed or semi-closed canopy were substantially reduced relative to open ground. Humidity was elevated. The accumulation of organic litter at the soil surface, leaves, woody debris, the remains of smaller plants, created a substrate layer of increasing biological complexity. Fungi, which had been present in terrestrial environments since at least the Ordovician, played a critical role in this environment. Mycorrhizal associations between fungal networks and plant root systems, documented from early Devonian fossil material, including specimens from the Rhynie Chert, facilitated mineral nutrient transfer between soil and plant tissue in ways that expanded the range of substrates plants could colonize and the efficiency with which they could do so. The geochemical consequences of forest expansion were large-scale and measurable. Forests drew down atmospheric carbon dioxide through photosynthesis at rates that open, sparsely vegetated landscapes could not approach. Carbon fixed in plant tissue, particularly in the lignin-rich woody structures that resisted decomposition, was increasingly removed from atmospheric circulation and transferred into long-term storage in soils and, eventually, sedimentary deposits. Current models of late Devonian atmospheric composition indicate a substantial decline in carbon dioxide concentrations across the period during which forests were expanding, with some estimates suggesting a reduction by a factor of several times relative to early Devonian levels. The climate implications of this drawdown was significant. Reduced greenhouse gas concentrations contributed to global cooling across the late Devonian, with evidence for glacial activity appearing in high-latitude deposits toward the end of the period. Root systems penetrating Devonian bedrock accelerated the chemical weathering of silicate minerals, a process that consumes carbon dioxide and releases calcium and magnesium ions into solution. Ions that ultimately reach the ocean via river systems and contribute to carbonate precipitation on the seafloor. The expansion of deep root systems across Devonian continents therefore amplified the carbon drawdown effect of photosynthesis through an additional geochemical pathway, connecting the biological activity of terrestrial forests directly to the chemistry of the marine environment thousands of kilometers away. The rivers draining forested late Devonian landscapes carried a different chemical and physical signature than those flowing across the barren early Devonian terrain. Increased organic carbon input from forest litter, altered nutrient delivery to coastal zones, and changes in sediment grain size and discharge patterns all reflected the transformation of the continental surface. These changes in riverine input to coastal marine environments contributed to the eutrophication. Excessive nutrient enrichment of shallow seas in some regions, with consequences for marine oxygen levels and ecosystem stability that would become increasingly significant toward the end of the period. The forests of the late Devonian were, by any measure, strange environments. The plants that composed them belonged to lineages now entirely extinct or represented only by dramatically reduced modern descendants. Their architecture was unfamiliar. Their ecological relationships, with fungi, with the earliest soil invertebrates beginning to colonize the litter layer, with the first vertebrates venturing into terrestrial margins, were being established for the first time, without precedent and without stability. Thick humidity gathered beneath canopies that had never before existed. Organic matter accumulated in soils that had never before contained it at any depth. Water moved differently across a landscape, now partially anchored by root systems that had not been there a few tens of millions of years before. The continents were changing, not quickly, by any human measure, but in geological terms with a speed that would leave a permanent mark on every system it touched. Atmospheric, oceanic, climatic, and biological. The age of bare rock and empty interiors was ending. Something was growing in its place, and the world it was building would never fully resemble what had come before. Where the land met the water, the rules changed. Not the rules of predation, those remained constant, operating with the same directness and finality they had always imposed. But the physical structure of the environment changed in ways that altered how predation worked, which animals were most vulnerable, and which strategies of ambush, pursuit, and evasion were most effective. The transitional zones between Devonian land and sea, river mouths, tidal flats, coastal swamps, flooded forest margins, were not simply geographic features. They were ecological filters, concentrating certain organisms, excluding others, and creating conditions that favored a specific and highly effective set of predatory strategies. The animals that evolved to exploit these zones were among the most specialized hunters of the Devonian world. The shallow water environments along Devonian coastlines and river systems were physically complex in ways that deeper marine environments were not. Water depth varied across short distances and changed seasonally, with rainfall and discharge patterns from river systems draining increasingly forested continental interiors. Submerged vegetation, primitive aquatic plants, and algal growth created structural complexity at and near the substrate. Organic material accumulating from terrestrial sources reduced water clarity through both particulate suspension and dissolved organic compounds that absorbed light. In many nearshore Devonian environments, effective visibility in the water column was measured in centimeters to low tens of centimeters rather than meters. These conditions systematically disadvantaged prey and advantaged ambush predators. An animal capable of remaining motionless against a substrate matching the visual texture of its surroundings and initiating a strike within a distance shorter than the prey's effective detection range operated with a structural advantage that open water pursuit predators did not possess. The geometry of shallow, visually constrained environments created, in effect, a persistent selective pressure toward ambush as the dominant predatory strategy. And the Devonian fossil record reflects this pressure in the anatomy and inferred behavior of several key lineages. Among the most consequential predators of Devonian shallow water and transitional environments were the rhizodont fishes, large lobe-finned predators within the broader Sarcoptean lineage whose fossil remains are known from late Devonian and subsequent Carboniferous deposits across multiple continents. Rhizodonts possessed large, recurved teeth with complex root structures. The feature from which the group takes its name set in robust jaws capable of securing large, struggling prey. The largest rhizodont taxa, including Rhizodus itself from Carboniferous deposits, reached estimated body lengths of 6 to 7 meters, though Devonian representatives were generally somewhat smaller. These animals inhabited freshwater and brackish environments, including the large river systems and flooded lowland areas that expanded across Devonian continental margins as forest cover increased and the hydrological character of the landscape changed. The ecological role of rhizodonts in Devonian freshwater systems was broadly analogous to that of large ambush predators in modern freshwater environments. Lying in wait in shallow, vegetated, low-visibility water and striking at fish, early tetrapods, and other vertebrates moving through their range. Their lobe-finned body plan with muscular pectoral and pelvic fins, capable of generating thrust and providing limited maneuverability in confined spaces, was well suited to operation in structurally complex shallow environments where the open water speed advantages of more streamlined fish were less relevant. The osteolepiform fishes represent another significant component of Devonian shallow water predator assemblages. Osteolepiforms were medium to large lobe-finned fish with robust bodies, large heads, and dentition structured for seizing and processing substantial prey. Eustopteran, one of the best-studied osteolepiform genera and known from exceptionally preserved material from the Miguasha locality in Quebec, Canada, reached body lengths of approximately 1 meter and possessed internal anatomy, including a vertebral column, skull structure, and a fin skeleton. That has made it one of the most referenced taxa for understanding the evolutionary transition toward tetrapods. Its preferred environment, inferred from the depositional context of Miguasha and comparable localities, was shallow, seasonally variable freshwater or estuarine. Exactly the transitional zone in which the selective pressures driving anatomical change toward terrestrial capability were most intense. In these environments, Eusthenopteran and related osteolipiforms would have operated as mid-level to upper predators, targeting smaller fish in low-visibility, structurally complex water.
Their ability to breathe air, inferred from the presence of internal nostrils and lung-like structures in closely related taxa,
would have provided a functional advantage in locally enriched waters of shallow Devonian swamps and river backwaters where dissolved oxygen concentrations could fall below the threshold for gill-based respiration alone. The interaction between these shallow water predators and the earliest tetrapods moving along Devonian water margins created a predation dynamic of particular intensity. Early tetrapods, transitioning between aquatic and terrestrial environments, were vulnerable in both directions. In the water, they lacked the speed and hydrodynamic efficiency of fully aquatic fish and were exposed to the ambush strategies of rhizodonts and large osteolepiforms operating in their preferred shallow zones. On land, they were slow, energetically constrained by the demands of weight-bearing locomotion in air, and limited in their ability to detect threats in an environment for which their sensory systems had not been fully refined. The marginal zone they occupied. The few meters of mudflat, tidal flat, and shallow water's edge between deep enough water to swim and dry enough land to walk was simultaneously their primary habitat and the zone of their greatest vulnerability. Evidence for predation on early tetrapods in Devonian environments is indirect but consistent with this picture. Bite marks on early tetrapod skeletal material, recovery of tetrapod remains in depositional contexts associated with large predatory fish, and the body size distributions of early tetrapod assemblages, which skew toward animals small enough to have used dense marginal vegetation as refuge. All suggest that predation pressure from aquatic predators was a persistent and significant selective force on early tetrapod populations. The shallow rivers and coastal swamps of the late Devonian were not safe corridors to the land. They were hunting grounds. Environments in which large, well-adapted predators had been operating for millions of years before the first tetrapods began to explore their margins. Every movement through shallow water, every crossing of an exposed mudflat, every return to the water's edge to feed or reproduce, brought early tetrapods into contact with ambushed predators whose anatomical refinement for exactly these conditions was, by that point, the product of a very long evolutionary history. Survival in these zones required more than the ability to move on land. It required the ability to navigate an environment where the water itself, the medium from which tetrapod life had emerged and to which it remained functionally tied, was also the primary source of lethal threat. That tension between dependency and danger, between the resource the water provided and the predators it concealed, shaped the early history of terrestrial vertebrate life in ways whose consequences extended far beyond the Devonian shoreline. The hunters in the shallow water were not simply predators. They were, in a measurable ecological sense, part of the force that drove vertebrate life onto land. Below the zones of active predation, below the reef margins and the shallow hunting grounds, below the turbulent interface where river systems discharged into coastal seas, the Devonian Ocean continued downward into environments that no large vertebrate regularly penetrated. These deeper zones were not empty. They were populated by organisms of extraordinary morphological diversity. Animals whose body plans, sensory systems, and ecological roles had been refined across hundreds of millions of years of marine evolution preceding the Devonian, and which continued to dominate the deeper and more stable portions of the ocean long after fish had come to define the shallow water fauna. To observe a Devonian seafloor community at depth would have been to encounter a biological world with almost no visual correspondence to anything familiar in the modern ocean. Not because the principles governing it were different, but because the specific organisms composing it belonged overwhelmingly to lineages that no longer exist. The Devonian was, in this sense, a world built from components that have since been almost entirely replaced. Trilobites, among the most recognizable of all Paleozoic invertebrates, were present in Devonian seas across a wide range of environments and ecological roles. Though their diversity had already passed its Ordovician peak by the time the Devonian began, Devonian trilobite faunas were nonetheless substantial, encompassing forms adapted to open water swimming, shallow reef-associated environments, and deeper benthic substrates. The trilobite body plan. A calcified dorsal exoskeleton divided into 3 longitudinal lobes and organized into a cephalon, thorax, and pygidium, conceals a diversity of functional anatomy that allowed different lineages to exploit substantially different ecological niches. Some Devonian trilobites possessed highly enlarged eyes with complex compound lens arrays, suggesting adaptation to low-light conditions in deeper water or turbid nearshore environments. Others show reduced or absent eyes, consistent with life in aphotic benthic zones where vision provided no advantage. The enrollment behavior of trilobites, the capacity to curl the body into a compact ball by flexing the thoracic segments until the head and tail shields met, is preserved in the fossil record and represents a defensive response to predation threat. The frequency with which enrolled trilobite specimens appear in Devonian deposits and the evidence for predatory damage on trilobite exoskeletons in the form of characteristic bite and crush marks indicates that predation pressure on trilobites was sustained and significant across the period. The predators responsible for this damage included fish with crushing dentition, nautiloid cephalopods, and potentially large eurypterids in shallower environments. The cephalopods of the Devonian seas represented a lineage already ancient by the period's beginning and diversifying with considerable speed across its duration. Nautiloid cephalopods, externally shelled forms broadly comparable in shell geometry to the modern nautilus, though belonging to entirely different families, had been major components of marine ecosystems since the Ordovician. By the Devonian, nautiloid diversity remained significant, with forms ranging from straight-shelled orthocones to tightly coiled geometries, and body sizes spanning from a few centimeters to, in some earlier lineages, several meters. These animals were active predators and scavengers, capable of jet propulsion through siphuncular water expulsion and equipped with grasping tentacles and a beak-like jaw structure capable of processing hard-shelled prey. More significant in the context of Devonian evolutionary history was the diversification of the ammonoids, a cephalopod group characterized by complex sutural patterns between the internal septae of the shell and the outer shell wall. Ammonoids first appear in the early Devonian fossil record and diversified rapidly across the period, establishing the lineage that would persist, with interruptions at major extinction boundaries. Until the end of the Cretaceous, Devonian ammonoid shells were generally modest in size relative to the forms that would characterize later periods, but their presence in Devonian deposits across multiple paleocontinental regions indicates rapid geographic expansion following the origin of the group, increasing across the group's evolutionary history, and is interpreted as providing increased resistance to hydrostatic pressure, potentially extending the depth range at which these animals could operate. The coral and stromatoporoid reef systems structured shallow Devonian marine environments, supported communities of sessile and mobile invertebrates of exceptional diversity. Brachiopods, marine invertebrates superficially resembling bivalve mollusks but belonging to an entirely separate phylum with a distinct internal anatomy, were among the most abundant and diverse components of Devonian reef, associated and shelf communities. Devonian brachiopod faunas included hundreds of documented species, occupying a range of substrate types and water depths, and filtering organic particles from the water column through a specialized feeding organ called the lophophore. Their calcified shells preserve with high fidelity in the fossil record, and Devonian brachiopod assemblages from well-studied localities provide detailed information about the composition, structure, and environmental distribution of benthic communities across the period. Crinoids, stalked echinoderms that filtered the water column through arrays of feathered arms radiating from a calyx mounted on a flexible stalk anchored to the substrate, formed dense aggregations on Devonian reef surfaces and in adjacent soft substrate environments. Crinoid gardens, visible in some Devonian limestone formations as closely packed accumulations of stalk and calyx fragments, indicate that these organisms achieved high population densities in favorable environments, constituting a significant component of the filter-feeding biomass in Devonian shelf seas and providing both habitat structure and food resources for the mobile invertebrates and fish that moved among them. In the water column above the bentho, planktonic communities of small organisms drifted through the photic zone, converting solar energy into organic matter through photosynthesis and providing the energetic foundation upon which all higher trophic levels ultimately depended. Acrotarchs, organic walled microfossils of uncertain and likely polyphyletic biological affinity, but interpreted in many cases as the remains of phytoplankton, are abundant in Devonian marine sediments and provide proxy information about surface ocean productivity and environmental conditions across the period. Graptolites, colonial organisms that had been major components of the Ordovician and Silurian plankton, were declining toward extinction through the Devonian, their ecological role in the plankton increasingly occupied by other groups. Chitinozoans, another group of organic walled microfossils present in Devonian sediments, are interpreted by some researchers as egg cases of marine invertebrates, and their distribution provides additional information about water mass conditions and oceanographic circulation patterns. The full vertical extent of the Devonian Ocean, from the photic surface waters through twilight zone and into the deeper aphotic basins, was therefore occupied by a continuous set of biological communities, each dependent on the layers above and below it for energy transfer, nutrient recycling, and the physical and chemical conditions necessary for survival. Nothing in this system was isolated. Every organism, from the photosynthetic plankton at the surface to the deposit-feeding invertebrates on the deepest successful seafloor, was connected to every other through chains of energy transfer and chemical exchange that operate and continue continuously and without interruption across the full depth of the water column. The Devonian Ocean was not a collection of separate environments. It was a single, vertically integrated system of extraordinary complexity, one that had taken hundreds of millions of years to assemble, and whose internal relationships were, by the late Devonian, more intricate and more interdependent than at any previous point in Earth's history. That interdependence, as would become clear, was also its vulnerability. Survival in any ecosystem is not a passive condition. It is not achieved by simply existing within an environment. It is maintained continuously and at metabolic cost against a background of competing demands for food, for space, for reproductive opportunity, for access to the physical and chemical conditions necessary to sustain biological function. In the Devonian, those demands were being negotiated simultaneously across land and sea. In ecosystems whose organizational complexity was, in many regions, approaching levels that had no clear precedent in the earlier fossil record, the organisms navigating these ecosystems were not simple. They were the products of hundreds of millions of years of evolutionary refinement, equipped with sensory systems, behavioral repertoires, and physiological tolerances shaped by the specific pressures of the environments they inhabited. But the environments themselves were changing, and the pace of that change, measured against the timescales of biological adaptation, was, in some intervals, uncomfortably fast. In the marine realm, Competition for space and food resources was structured primarily by the architecture of the reef systems that dominated shallow tropical and subtropical seafloors across the Middle and Late Devonian. Reef environments concentrated biological activity in spatially defined zones where the substrate provided attachment points for sessile filter feeders, refuge for mobile invertebrates, and hunting ground for predatory fish. Space on and near the reef surface was a finite resource, and competition for it among sessile organisms – corals, stromatoporoids, brachiopods, crinoids – was intense and continuous. Overgrowth competition, in which faster-growing organisms hailed slower-growing neighbors, was a primary mechanism of competitive exclusion in reef communities, and the skeletal record of Devonian reef formations preserves evidence of this process in the form of encrusting growth patterns and the spatial relationships between adjacent colonial organisms. Mobile organisms within reef environments competed for food resources distributed unevenly across the reef structure. Patch reefs surrounded by soft substrate, isolated from other reef structures, supported communities whose composition reflected the balance between local productivity and the predation pressure exerted by resident and transient predators. Fish species partitioned these environments by feeding strategy, some grazing on algae and sessile invertebrates, others targeting mobile invertebrate prey within the reef structure, others hunting smaller fish in the water column above the reef crest. The degree of ecological specialization visible in Devonian fish faunas from reef-associated localities indicates that niche differentiations had proceeded to a considerable depth by the late Devonian, with multiple species occupying the same reef environment by exploiting different resources or operating in different microhabitats. Oxygen availability was a pervasive and spatially variable constraint on survival in Devonian aquatic environments. In well-mixed, wave-agitated shallow water above reef structures, dissolved oxygen concentrations were generally sufficient to support the full range of aerobic metabolism. But in deeper basinal environments, in the bottom waters of stratified water columns, and in the organically enriched backwaters of river systems draining heavily vegetated late Devonian landscapes, oxygen concentrations could fall to levels that excluded most metazoan life. These low oxygen zones, documented in Devonian sedimentary sequences by the presence of finely laminated organic-rich mudstones lacking evidence of bioturbation by burrowing organisms, represented barriers to the distribution of aerobically respiring animals and created refuges of a kind for organisms tolerant of reduced oxygen conditions. The expansion of low-oxygen bottom waters in some Devonian basins across the late Devonian was connected, through geochemical pathways, to the increasing input of terrestrially derived organic material and nutrients to coastal seas. Forest cover expanded across the continents. Elevated nutrient delivery stimulated surface productivity, increasing the flux of organic matter sinking through the water column. Bacterial decomposition of this organic matter consumed dissolved oxygen in deeper water, expanding the volume of oxygen-depleted bottom environments. This process, broadly comparable to the eutrophication events documented in modern coastal seas, receiving high nutrient input from agricultural landscapes, operated on a geological timescale in the Devonian, but produced measurable changes in marine oxygen distribution that directly constrained the survival options available to bottom-dwelling and deep-water organisms. On land, the survival challenges facing early tetrapods and the arthropods that had preceded them into terrestrial environments were of a fundamentally different character. The primary constraints were physiological. Water loss through exposed body surfaces was an immediate and continuous threat for any organism operating in air rather than water. Early tetrapods whose skin lacked the keratinized barrier that characterizes the integument of later fully terrestrial vertebrates were restricted to environments where ambient humidity was high enough to limit desiccation to tolerable rates. This constraint confined them, functionally, to the margins of water bodies. the same transitional zones that also placed them within range of the aquatic predators hunting in shallow water. Locomotion on land imposed energetic costs that had no equivalent in aquatic environments. In water, buoyancy offsets the effects of gravity on body mass, and forward movement through a fluid medium is primarily constrained by drag rather than the weight-bearing demands imposed on limbs by gravity in air. On land, the full weight of the body is transferred through the limbs to the substrate at each step, requiring musculoskeletal structures capable of supporting that load through the range of motion involved in locomotion. The limb anatomy of early tetrapods, evolved initially for function in water, was not optimally configured for this demand, and the energetic cost of terrestrial movement in early tetrapods was likely substantially higher per unit distance than in later, more derived terrestrial vertebrates whose limb geometry had been refined under sustained selection for efficient weight-bearing locomotion. Food acquisition on land presented its own challenges. The arthropod prevailable in Devonian terrestrial environments. Early myriapods, arachnids, and the earliest insects appearing toward the end of the period were present but not uniformly distributed, locating and capturing prey in a three-dimensional terrestrial environment using sensory systems calibrated primarily for function in water, required behavioral and neurological adjustments whose pace of development is difficult to assess from the fossil record alone, but whose necessity is implied by the anatomical evidence of the transition. Climate instability added a further dimension of unpredictability to survival across Devonian ecosystems. Evidence from the sedimentary and geochemical record indicates that the Devonian was not a period of stable, uniform climate. Sea level fluctuated repeatedly across the period, reflecting changes in ice volume, tectonic activity, and oceanic heat distribution. Periods of reef growth and carbonate deposition alternated with intervals of reduced reef development and increased siliciclastic input, recording shifts in environmental conditions that affected reef communities on timescales of tens of thousands to millions of years. Storm frequency and intensity, recorded in sedimentary event layers, varied across the period and imposed episodic physical disturbance on shallow marine communities that added unpredictability to an already demanding survival environment. The climate changes associated with the drawdown of atmospheric carbon dioxide by expanding forests. Cooling temperatures, shifting precipitation patterns, that altered the conditions available to early land-dwelling organisms across the late Devonian in ways whose biological consequences are only partially resolved by the available fossil record. Survival across all of these environments and under all of these pressures was never guaranteed. It was an outcome produced, generation by organisms whose inherited biology was adequate, barely, in many cases, to the demands of the environment they occupied. Those whose biology was not adequate left no descendants. Those whose biology was sufficient left the fossil record that makes it possible, hundreds of millions of years later, to reconstruct the world in which they lived and died. Above the forests and the shallow seas, above the mudflats and the reef margins, the Devonian atmosphere was a physical environment in its own right. Not empty space, but a chemically active, dynamically structured system whose properties shaped every biological process occurring beneath it. The air of the Devonian was not the air of the modern world. Its composition was measurably different. Its optical properties, the way it transmitted, scattered and absorbed radiation across the electromagnetic spectrum, were distinct from those of the modern atmosphere in ways that had direct consequences for surface temperature, biological exposure to ultraviolet radiation, and the energy available to drive photosynthesis at the land surface. Understanding what existed above the Devonian landscape requires understanding the atmosphere not as background, but as a participant, a system actively involved in the biological and geological transformations occurring beneath it. Oxygen concentrations in the early Devonian atmosphere are estimated from geochemical proxy data and carbon cycle modeling to have been in the range of 15 to 16% by volume, meaningfully lower than the approximately 21% characterizing the modern atmosphere. This difference had several categories of physical and biological consequence. At lower oxygen partial pressure, the photochemical production of stratospheric ozone, generated by the interaction of ultraviolet radiation with oxygen molecules, proceeds at a reduced rate. The ozone layer provides the primary shield against high-energy ultraviolet B and ultraviolet C radiation reaching Earth's surface. A thinner ozone column, resulting from lower atmospheric oxygen, would have allowed greater ultraviolet flux at the surface than modern organisms at equivalent latitudes experience. For organisms attempting to colonize exposed terrestrial surfaces, early vascular plants, the first sauropods moving onto land, the earliest tetrapods venturing across open mudflats. This elevated ultraviolet environment represented a direct physiological stress capable of damaging nucleic acids and proteins in exposed tissue and imposing selection pressure toward protective pigmentation, behavioral avoidance of direct sun exposure, or confinement to habitats where water, dense vegetation, or substrate provided natural shielding. The relationship between atmospheric oxygen, ozone production, and surface ultraviolet flux was not static across the Devonian. As vascular plant cover expanded across the continents through the Middle and Late Devonian, photosynthetic oxygen production increased. Current atmospheric models indicate that oxygen concentrations rose progressively across the later Devonian, approaching and possibly exceeding modern levels in the Carboniferous. This rise in oxygen had consequences for ozone abundance, surface ultraviolet flux, and the combustibility of terrestrial vegetation. The last of these, a factor that would become increasingly significant as forest biomass accumulated across late Devonian and Carboniferous landscapes. Carbon dioxide concentrations in the Devonian atmosphere were substantially elevated relative to modern pre-industrial values. Estimates from multiple proxy methods, including analysis of paleosol carbonate isotopes, stomatal density in fossil plant material, and carbon cycle modeling, suggest early Devonian carbon dioxide concentrations in the range of 10 to 15 times modern pre-industrial levels, declining substantially across the period as forest expansion drove increased carbon sequestration. So, this elevated carbon dioxide environment had direct effects on the global energy balance. Enhanced greenhouse, forcing maintaining surface temperatures warmer than modern values at equivalent solar luminosity, and indirect effects on plant physiology, since higher ambient carbon dioxide concentrations influence the efficiency of photosynthesis and the degree to which plants open their stomata to exchange gases with the atmosphere. The decline in atmospheric carbon dioxide across the Middle and Late Devonian, driven primarily by the expansion of forest cover and the associated increase in silicate weathering rates driven by deep root systems, represents one of the most significant geochemical perturbations of the Phanerozoic. The rate of this decline and its consequences for global temperature and ocean chemistry are active areas of research. But from high carbon dioxide warmth in the early Devonian toward cooler conditions in the late Devonian and Carboniferous is established across multiple independent proxy records. The weather systems operating over Devonian continents and oceans were driven by the same fundamental physical principles governing modern atmospheric circulation, but their specific patterns reflected the different continental configurations, sea surface temperatures, and land surface properties of the Devonian world. The shallow epicontinental seas that covered large portions of the Devonian continental interior provided extensive low-albedo water surfaces that absorbed solar radiation and transferred heat to the overlying atmosphere through evaporation and sensible heat flux. These inland seas moderated continental temperatures in adjacent regions, reducing the seasonal temperature extremes that would have characterized fully continental interiors and supplied brief moisture to atmospheric circulations systems that redistributed it as precipitation across the landscape. Storm systems over Devonian shallow seas were likely frequent and, in some intervals, intense. The thermal contrast between warm tropical ocean surfaces and the cooler air masses descending from higher latitudes drove convective activity in tropical and subtropical regions, producing precipitation events capable of generating significant runoff across partially vegetated continental surfaces. In the early Devonian, before root systems had stabilized terrestrial substrates, rainfall events of sufficient intensity would have mobilized large volumes of sediment, contributing to the high sediment loads documented in Devonian river system deposits and the turbid conditions inferred for many Devonian nearshore marine environments. Later, as forest cover expanded through the Middle and Late Devonian, the physical properties of the land surface changed in ways that altered atmospheric circulation at regional scales. Forested surfaces have higher roughness length than bare substrate, increasing the turbulent exchange of momentum, heat, and moisture between the surface and the overlying atmosphere. Transpiration from forest canopies returns water vapor to the atmosphere at rates substantially higher than evaporation from bare soil, increasing regional precipitation recycling and stabilizing moisture availability across forested landscapes. The progressive greening of Devonian continents was therefore not simply a biological event. It was a change in the physical boundary conditions of the atmosphere, altering the inputs to weather systems that in turn influenced the distribution of precipitation, the frequency of drought, and the stability of the terrestrial environments in which biological communities were establishing themselves for the first time together. Above the highest reaches of weather systems, the stratosphere and upper atmosphere continue to interact with solar and cosmic radiation in ways that influenced the surface environment on longer timescales. Variations in solar output across the Devonian, while not directly measurable, are expected to have followed the gradual brightening trend characterizing solar evolution across geological time. The sun of the Devonian delivering slightly less total radiation than the modern sun, a difference partially offset by the stronger greenhouse forcing of the higher carbon dioxide Devonian atmosphere. Reduced solar input and elevated greenhouse warming is one component of the broader climate system whose behavior across the Devonian is still being refined through the integration of proxy data and modeling approaches. The atmosphere above the Devonian world was not passive background, carrying heat from the tropics toward the poles, redistributing moisture across continental surfaces, filtering the radiation reaching living tissue, and responding continuously to the changes occurring at the surface beneath it. As forests grew, the atmosphere changed. As the atmosphere changed, the climate shifted. As the climate shifted, the ecosystems that had assembled themselves under one set of conditions were required to persist under another. That requirement to survive within a system that did not remain constant was not unique to the Devonian, but the Devonian was among the first periods in Earth's history in which biological activity had become a primary driver of the atmospheric change, to which life itself then had to respond. The sky above a primitive Earth was already, in this sense, a mirror of the life growing beneath it. The rivers of the Devonian world were not simple conduits for water moving from high ground to sea. They were ecosystems in their own right, dynamic, physically complex, and biologically dense in ways that reflected both the productivity of the aquatic environments they connected and the increasing biological complexity of the terrestrial landscapes through which they moved. As forest cover expanded across Devonian continents through the Middle and Late Devonian, the character of river systems changed in measurable ways, and those changes cascaded through every biological community dependent on freshwater environments for survival or the transit between aquatic and terrestrial life. The rivers of the early Devonian, flowing across largely unvegetated substrates, were broad, shallow, and heavily loaded with sediment. Their channels migrated frequently across wide, unstable floodplains, shifting course in response to discharge variations and the low cohesion of unbound substrate. As root systems began to penetrate and stabilize banks through the Middle Devonian, channel geometry changed. Stabilized banks resisted lateral migration. Channels became more confined, deeper relative to their width, and capable of maintaining more consistent flow paths across the floodplain. The transition from braided to meandering channel geometry, recorded in Devonian sedimentary sequences at multiple localities, reflects this biological influence on physical river dynamics. One of the earliest documented instances of life directly modifying the physical behavior of a major Earth surface system. The biological communities within Devonian river systems were structured by the same fundamental ecological principles operating in marine environments, but expressed through the specific physical and chemical characteristics of freshwater. Dissolved oxygen in freshwater systems is influenced by temperature, flow velocity, and organic load. Fast-moving, well-oxygenated headwater streams supported communities different from those in slow-moving, organic-rich lowland reaches. In the lowland floodplain environments that expanded as forest cover increased and organic input to river systems grew, dissolved oxygen concentrations in bottom waters and in the slower-moving portions of the channel could fall to levels that stressed aerobically respiring organisms, particularly during warm seasons when oxygen solubility in water decreases and bacterial decomposition of accumulated organic matter accelerates. These low oxygen conditions in portions of Devonian floodplain river systems created selection pressure for exactly the kind of aerial respiration supplementation documented in several Devonian fish lineages. The lungfish. Members of the group Dipnoi, which had diversified through the Devonian, and whose living representatives retain functional lungs capable of aerial respiration, were particularly well adapted to periodically oxygen-depleted freshwater environments. Devonian lungfish are known from deposits across multiple continents, and their distribution in Frere Oish and Estuarine sedimentary sequences indicates a broad tolerance for the variable oxygen conditions characteristic of Devonian floodplain river systems. Some lungfish lineages show evidence of estivation, the capacity to survive dry seasons by burrowing into substrate and entering a state of metabolic depression, a strategy directly relevant to survival in seasonally variable Devonian river environments where water availability itself was not constant. The predators inhabiting Devonian river systems were drawn from lineages adapted to the specific physical constraints of freshwater environments. Large rhizodont lobe-finned fish, as noted in the context of coastal transitional environments, were also significant components of Devonian freshwater predator assemblages. Their robust bodies, large recurved teeth, and muscular lobe fins made them effective ambush predators in the shallow, structurally complex environments of floodplain rivers and oxbow lakes. The isolated, curved water bodies that form when meandering river channels cut off and abandon sections of their former course. Oxbow lakes, increasing in frequency as river channels became more stable and meandering through the late Devonian, would have represented concentrated prey environments. Isolated bodies of water in which fish populations were effectively trapped, unable to disperse across the surrounding landscape, and subject to predation pressure from any large predator capable of accessing the oxbow through connected channels or across short sections of intervening floodplain during high water events. The seasonal flooding of Devonian floodplains created additional ecological dynamics of direct relevance to both predators and prey. As river discharge increased during wet seasons, water spread across low-lying floodplain surfaces, inundating areas of terrestrial habitat and creating temporary aquatic environments of enormous lateral extent but shallow depth. These flooded floodplain surfaces concentrated fish moving out of the main channel to exploit food resources, invertebrates, plant material, and the organic detritus accumulating in terrestrial environments. that became accessible during inundation. Predators capable of following prey into these shallow, flooded environments and of navigating effectively in water only a few centimeters to perhaps a meter in depth had access to prey concentrations unavailable in the main channel. Early tetrapods moving along floodplain margins during inundation events were simultaneously exploiting these temporary aquatic resources and exposing themselves to the predators tracking the same prey concentrations into the same shallow environments. The flooded Devonian floodplain was therefore not a refuge from aquatic predation, but an extension of it, a temporary aquatic environment in which the structural constraints on ambush predation were, if anything, more favorable to predators than the deeper, more open waters of the main channel. The geochemical role of Devonian rivers in connecting terrestrial and marine environments extended beyond the physical transport of sediment and water. Dissolved nutrients, particularly phosphorus and nitrogen released from weathering rock and decomposing organic material in forest soils, were carried by river systems to coastal marine environments, where they stimulated primary productivity in the photic zone. Was not constant. It varied with discharge, with the extent of forest cover in the catchment, and with the degree of soil development in the terrestrial environment. During periods of high discharge following major rainfall events, nutrient pulses delivered to coastal seas could stimulate algal blooms of sufficient intensity to affect dissolved oxygen concentrations in the bottom waters of adjacent shallow marine basins. The connection between terrestrial forest expansion, increased soil nutrient release, elevated river nutrient discharge, and marine eutrophication represents one of the most significant biogeochemical linkages of the late Devonian. A chain of cause and effect connecting the biological transformation of continental surfaces to the chemical conditions of marine environments thousands of kilometers from the nearest forest. The rivers carried more than water and sediment. They carried the chemical signature of a changing land surface. As the dissolved products of biological activity in soils that had not existed a few tens of millions of years earlier, and delivered that signature to marine ecosystems that no evolutionary preparation for receiving it at the concentrations as rates the expanding Devonian forest was beginning to generate. The rivers of the Devonian were alive, and what they carried downstream was already beginning to change the sea. Evolution does not proceed toward a destination. It has no trajectory, no preferred outcome, no internal logic directing biological change toward complexity, efficiency, or any other property that might, from a human perspective, seem like progress. What evolution produces at any given moment in geological time is simply the set of organisms whose inherited characteristics were adequate in the specific environments they occupied under the specific pressures those environments imposed to survive long enough to reproduce. The Devonian fossil record, examined across its full temporal and geographic extent, reflects this process operating across an extraordinary range of lineages, simultaneously generating biological diversity of a kind and at a rate that transformed the character of life on Earth within the span of approximately 60 million years. That transformation was not smooth. It was not uniform. It proceeded at different rates in different lineages, produced dead ends as frequently as it produced innovations that persisted, and was shaped at every point by urgency the specific environmental conditions, competitive pressures, and chance that determined which variants survived and which did not. The diversification of jawed vertebrates across the Devonian represents one of the most rapid and consequential episodes of evolutionary radiation in the history of animal life. At the beginning of the Devonian, jawed fish were present but not dominant. The seas still contained abundant jawless fish. Agnathans, including heavily armored forms such as the heterostracans and osteostracans, alongside the early placoderms and the first representatives of the Chondrichthyan and Osteichthyan lineages, By the end of the landscape of vertebrate diversity had been almost entirely restructured. Agnathans were reduced to the two surviving lineages, lampreys and hagfish, that persist today in substantially modified form. Placoderms, dominant through most of the period, would not survive the end Devonian extinction, the lineages that would define vertebrate diversity in all subsequent periods. Chondrichthyans and Osteichthyans, including the lobe-finned fish from which tetrapods descended, had diversified to the point where their ecological dominance was effectively established before the Devonian closed. The speed of this restructuring, measured against the backdrop of geological time, was significant. The mechanisms driving it included competitive displacement, in which diversifying jawed fish outcompeted jawless fish for shared resources, as well as the opening of new ecological niches as reef systems expanded, terrestrial environments became colonizable, and the physical structure of marine and freshwater habitats grew more complex. The jaw itself, derived from modified anterior gill arch elements, was a mechanical innovation of enormous ecological consequence. It expanded the range of food items accessible to the animals possessing it, enabled predation strategies unavailable to filter feeders and suction feeders, restricted to small, soft-bodied prey, and provided the structural foundation on which an enormous variety of subsequent dental and cranial specializations were built. The evolution of sensory systems across Devonian vertebrates tracks the diversification of ecological roles with considerable fidelity. Vision in Devonian fish shows evidence of increasing sophistication in lineages associated with active predation in well-lit environments. The large orbits and inferred large eyes of some predatory placoderms and early bony fish suggest investment in visual acuity consistent with visually guided prey detection and pursuit. In contrast, lineages associated with deeper water, burrowing, or highly turbid environments show reduced orbital size or evidence of reliance on non-visual sensory modalities. The lateral line system, which detects water movement and pressure gradients, and the electroreceptive system, which detects bioelectric fields generated by the muscular activity of nearby organisms. The lateral line system, present across a wide range of Devonian fish lineages, was particularly significant in the low-visibility environments that characterized much of the Devonian coastal and estuarine zone. In water where effective visual range was measured in centimeters, the capacity to detect the pressure waves generated by a moving organism at distances of tens of centimeters to meters represented a meaningful sensory advantage for predators attempting to locate prey, and for prey attempting to detect the approach of predators. The evolutionary arms race between predator sensory capability and prey evasion behavior in Devonian low-visibility environments drove refinement of the lateral line system in multiple lineages simultaneously, producing convergent sensory solutions to shared environmental problems across phylogenetically distant groups. The evolution of armor in Devonian fish lineages illustrates the same principle from the defensive direction. Dermal, bone. Bone forming in the skin, rather than from cartilaginous precursors, provided structural protection against predation in numerous Devonian fish lineages independently. In placoderms, the dermal armor of the head and trunk shield was the defining anatomical feature of the group. In early actinopterygian fish, the ray-finned fish whose descendants would eventually come to dominate aquatic environments globally, thick ganoid scales provided a flexible but resistant surface covering. In some jawless fish persisting into the Devonian, elaborate dermal armor covered extensive body regions. The repeated, independent evolution of dermal protection across Devonian fish groups reflects the intensity of predation pressure in these environments, a pressure so consistent and so directional that similar defensive solutions emerged in lineages with no close common ancestor possessing such structures. The metabolic cost of producing and maintaining dermal armor is not negligible. Bone requires calcium, phosphorus, and the sustained cellular activity of osteoblasts and osteoclasts across the animal's lifetime. An organism investing resources in armor production has fewer resources available for growth, reproduction, and other functions. The prevalence of armor in Devonian fish lineages therefore indicates that the selective benefit of protection from predation, measured in survival probability and reproductive success, consistently exceeded this metabolic cost across the environments in which armored lineages lived, where predation pressure was reduced, as in some deeper or more isolated environments, armor tended to be reduced correspondingly, reflecting the metabolic savings available when the cost of protection outweighed its benefit. The evolution of reproductive strategies across Devonian vertebrates added further dimensions to the diversification of the group. Evidence from exceptionally preserved placoderm specimens, including material from the Gogo Formation in Western Australia, where three-dimensional soft tissue preservation has provided information unavailable from most Devonian localities, indicates that some placoderm lineages had evolved viviparity, the retention of developing embryozoos within the mother's body and their nourishment through this reproductive strategy, which provides developing offspring with protection from predation and environmental stress at the cost of significant maternal resource investment, implies a level of physiological complexity in some Devonian placoderms that substantially revise early assumptions about the reproductive biology of early jawed vertebrates. The coexistence of viviparous placoderms with egg-laying lineages in the same Devonian environments indicates that multiple reproductive strategies were viable simultaneously. That no single approach to pre-production was uniformly superior across the range of conditions present in Devonian seas. This diversity of reproductive biology mirrors the broader pattern of Devonian vertebrate evolution, not convergence toward a single optimal solution, but the maintenance of multiple functional strategies under conditions varied enough that different approaches remained selectively viable in different contexts. The Devonian was not producing a single kind of fish refined toward perfection. It was producing dozens of experiments simultaneously. In jaw mechanics, in sensory capability, in armor configuration, in reproductive strategy, in locomotion and habitat use, most of which would eventually be discontinued, and a small number of which would persist long enough to become the ancestors of everything that came after. That is what evolution looks like, measured across geological time. Not progress, not direction, a vast, continuous, undirected process of variation and selection, generating, in the Devonian, more biological novelty per unit time than almost any comparable interval in the history of vertebrate life. The signs were not sudden. They rarely are in the geological record of major environmental transitions. What the sedimentary and geochemical record of the late Devonian preserves is not a single catastrophic event, but a prolonged interval of increasing instability, period during which the environmental conditions that had supported the extraordinary biological complexity of Devonian ecosystems began at multiple levels and through multiple mechanisms. The collapse was not instantaneous. It was structured, a sequence of pulses, each one stressing the system further, each one reducing the capacity of surviving communities to absorb the next disturbance. Understanding why the Devonian ended the way it did requires understanding the system that was failing, not as a collection of individual species disappearing one by one, but as an integrated network of biological and geochemical relationships whose internal connections made the failure of any one component capable of propagating
consequences through the rest.
The marine oxygen crisis of the late Devonian is among the most extensively documented environmental changes of the period, and it is recorded across multiple geographic regions in consistent and mutually reinforcing proxy data. Black shales, fine-grained sedimentary rocks rich in organic carbon, deposited in bottom waters too oxygen depleted to support the burrowing organisms whose bioturbation would otherwise have disrupted laminated sediment structure, appear with increasing frequency and geographic extent in late Devonian stratigraphic sequences. The Kelvasser events, two discrete intervals of widespread anoxic deposition identified within the Frasnian stage of the late Devonian, are recognized in sedimentary sequences across North America, Europe, and North Africa, indicating that oxygen depletion in marine bottom waters was not a local phenomenon but a global-scale perturbation affecting the chemistry of ocean basins across multiple paleocontinental regions simultaneously. The mechanisms driving this oxygen depletion were not simple. Current models implicate a combination of factors operating through interconnected pathways. The expansion of forest cover across late Devonian continents increased the delivery of organic carbon and bioavailable nutrients, particularly phosphorus, released from accelerated silicate weathering driven by deep root systems to coastal marine environments via river discharge. Elevated nutrient input stimulated surface ocean productivity, increasing the flux of organic matter sinking through the water column. Bacterial decomposition of this organic matter at depth consumed dissolved oxygen, expanding the volume of anoxic bottom water. In stratified water columns where mixing between surface and deep water was limited, the rate of oxygen consumption at depth could exceed the rate of resupply from the surface, driving persistent anoxia across basin floors and lower slopes. The reef systems of the Devonian are among the most biologically complex and geographically extensive in Earth's history, showed measurable signs of stress before the major extinction pulses of the Frasnian-Furmenian boundary. Reef growth requires specific environmental conditions, water temperatures within a tolerable range, carbonate light penetration, and carbonate saturation states sufficient to support skeletal construction that interfere with the physiology of reef-building organisms. As late Devonian oceanographic conditions shifted, driven by the combination of cooling surface temperatures, altered nutrient dynamics, and expanding bottom water anoxia, the window of environmental conditions within which Devonian reef builders could function narrowed progressively. Evidence for reef stress in the late Devonian includes changes in reef community composition preceding the major extinction events. Shifts in the relative abundance of different reef-building organisms, reductions in reef accretion rates, and intervals of reef drowning in which carbonate production ceased before being resumed under temporarily improved conditions. These pre-extinction indicators suggest that the reef systems were not simply destroyed abruptly by a sudden environmental catastrophe, but were weakened progressively across an extended interval. Their resilience reduced by repeated stress events before the conditions of the Frasnian-Furmenian boundary delivered impacts they could not recover from. The loss of reef systems had cascading consequences throughout the marine ecosystem. Reefs were not simply physical structures. They were biological engines that generated habitat complexity, concentrated productivity, and supported the enormous diversity of organisms dependent on reef architecture for food, shelter, and reproductive opportunity. As reefs deteriorated, the communities they supported fragmented. Species whose survival depended on reef-associated habitat lost the environmental infrastructure their biology required. The diversity of shallow marine environments declined not simply because species were dying directly from anoxia or temperature stress, but because the habitat that had organized and sustained their communities was disappearing. Volcanic activity contributed additional complexity to the late Devonian environmental system. Large igneous province emplacement. The eruption of enormous volumes of basaltic magma across geologically brief intervals has been proposed as a contributing factor to late Devonian environmental instability, with the Vilu traps of Siberia dating to the late Devonian, identified as a potential source of volcanic gases capable of perturbing atmospheric composition and ocean chemistry. Volcanic outgassing of carbon dioxide, sulfur dioxide, and other compounds can influence global temperature, ocean acidification, and the delivery of volcanic ash and associated chemical compounds to marine environments. The precise timing and magnitude of Veliotrap's activity relative to the Devonian extinction pulses remains an area of active research, and the degree to which volcanism contributed to, rather than coincided with, the environmental deterioration of the late Devonian is not fully resolved. What is established is that the late Devonian environmental system was subject to multiple simultaneous perturbations, from the geochemical consequences of forest expansion, from oceanographic changes associated with cooling and altered circulation, from the expansion of marine anoxia, and potentially from volcanic forcing, whose combined effect on the stability of marine ecosystems was substantially greater than any single factor would have produced in isolation. The response of marine communities to this accumulating environmental stress was visible in the fossil record well before the major extinction pulses that define the Frasnian-Furmenian boundary. Diversity indices for shallow marine invertebrate communities showed declining trends in the later Frasnian, with richness reduced and community composition shifting toward assemblages dominated by stress-tolerant generalists, rather than the specialized reef-associated taxa that had characterized peak Devonian marine diversity. The geographic ranges of some taxa contracted before their final disappearance from the fossil record, as the environmental conditions they required became spatially restricted to refugia of adequate oxygen, temperature, and substrate quality, coral lineages that had built the great reef systems of the Middle Devonian. The tabulate and rugose corals, whose skeletal remains form the structural core of Devonian reef limestones of rock across multiple continents, show evidence of declining diversity and reef-building capacity through the late Frasnian. Stromatoporoids, the sponge-grade organisms that had been among the most important reef constructors of the Devonian, similarly declined. The reef ecosystem that had organized shallow marine environments for tens of millions of years was losing the organisms that built it, not instantaneously, but progressively across an interval long enough to be resolved in the sedimentary record as a trend rather than an event. The communities it had supported for tens of millions of years were losing the environmental conditions that had made them possible, and the process, once begun, did not reverse. The end came in pulses, not in a single catastrophic moment, not in the instantaneous collapse that popular accounts of mass extinction sometimes imply, but in a sequence of discrete environmental crises separated by intervals of partial recovery. Intervals during which surviving communities began to reorganize, reef systems showed tentative signs of renewed growth, and diversity began to recover before the next pulse arrived, and compressed it again. The extinction that closes the Devonian period is formally identified with the Frasnian-Furmenian boundary, approximately 372 million years ago, and with a second significant pulse near the end of the Furmenian stage, approximately 359 million years ago, the latter marking the boundary between the Devonian and the Carboniferous period. Together, these events constitute what is recognized in the paleontological literature as one of the 5 major mass extinctions of the Phanerozoic eon, an interval of biological loss comparable in magnitude, if not in mechanism, to the end Permian and end Cretaceous events that receive considerably more public attention. The scale of the loss was substantial. Current estimates, derived from analysis of the fossil record across multiple taxonomic groups and geographic regions, suggest that somewhere between 70 and 80% of all marine species present in the late Devonian did not survive into the Carboniferous. The loss was not distributed uniformly across ecological groups. It was selective in ways that reflect the specific mechanisms of environmental deterioration operating through the extinction interval, and that selectivity tells a story about what those mechanisms were and how they operated. The organisms most severely affected by the Devonian extinction were those most dependent on the specific environmental conditions of shallow, warm, well-oxygenated, tropical and subtropical marine environments. Reef-building organisms, The tabulate and rugose corals, the stromatoporoids, were effectively eliminated as ecologically significant reef constructors. The great reef systems of the Devonian, some of the most extensive in Earth's history, collapsed across the Frasnian-Furmenian boundary and did not recover in any comparable form for approximately 10 to 15 million years into the Carboniferous, an interval referred to in the geological literature as the reef gap, during which carbonate reef construction was essentially absent from the global marine record. The organisms that eventually rebuilt reef systems in the Carboniferous belonged to different lineages, operating through different biological mechanisms, producing structures with different ecological properties than the Devonian reefs they replaced. Brachiopods, among the most diverse and abundant components of Devonian shallow marine communities, suffered severe diversity loss across the extinction interval. Many of the reef-associated brachiopod lineages that had characterized Middle Devonian faunal assemblages did not survive into the Furmenian. Those that did were disproportionately represented by forms tolerant of a wider range of environmental conditions. Organisms whose biology was less narrowly calibrated to the specific parameters of warm, clear, well-oxygenated shallow water. Trilobites, already reduced from their Ordovician diversity peak, experienced further significant losses across the Devonian extinction interval. Several trilobite orders that had persisted through the Devonian did not survive into the Carboniferous. The lineages that continued were, again, those whose ecological tolerances extended across a broader range of conditions than the specialists most vulnerable to the environmental changes of the late Devonian. The armored fish that had dominated Devonian seas as the apex predators and occupied a wide range of ecological niches across the period did not survive the end Devonian extinction. This is among the most consequential facts of the Devonian biological record. The placoderms had been the dominant, jawed vertebrates of the Devonian across virtually every ecological role occupied by jawed fish: apex, predation, mid-level predation, benthic feeding, filter feeding, and open water pursuit. Their diversity had been extraordinary. More than 350 genera are currently recognized in the fossil record, distributed across all major marine and freshwater environments of the Devonian world. And yet not a single placoderm lineage is found in Carboniferous deposits. The group was entirely eliminated. The mechanism of placoderm extinction is not fully resolved. The end Devonian environmental changes: expanding marine anoxia, reef collapse, cooling of surface waters, altered food web structure following the loss of reef-associated productivity, would have imposed stress across multiple trophic levels simultaneously. Apex predators, dependent on the productivity of lower trophic levels for their food supply, are often disproportionately vulnerable to ecosystem-level disruptions that reduce the abundance and diversity of prey. The collapse of reef systems and the loss of the diverse shallow marine communities those systems supported would have reduced the prey base available to large placoderm predators, potentially below the threshold necessary to sustain viable populations across their former geographic range. The vacancy left by placoderm extinction was not immediately filled. The early Carboniferous records a period of reduced marine vertebrate diversity, a gap in the fossil record of jawed fish sometimes referred to as Romer's Gap, during which the lineages that would eventually come to dominate post-Devonian seas were present but not yet ecologically significant. Sharks, which had coexisted with placoderms through the Devonian without achieving ecological dominance, diversified substantially in the aftermath of the extinction, eventually occupying the apex predator roles that placoderms had vacated. The transition was not instantaneous, and the Carboniferous oceans of the interval immediately following the Devonian extinction were ecologically impoverished relative to the systems they replaced. The terrestrial record of the end-Devonian extinction is less completely documented than the marine record, in part because terrestrial preservation is inherently less continuous and less geographically uniform than marine sedimentation. Evidence from the plant fossil record indicates significant floral changes across the Devonian Carboniferous boundary, with some lineages reduced and others expanding into the ecological space created by the loss of competitors. The early tetrapods that had been diversifying cautiously through the late Devonian, moving along water margins, exploiting the transitional zone between aquatic and terrestrial environments, survived the end Devonian extinction and diversified substantially in the Carboniferous, eventually giving rise to the lineages that would colonize terrestrial environments fully and permanently. Their survival, through an extinction that eliminated the majority of marine vertebrate diversity, reflects the degree to which the terrestrial marginal, demanding, and biologically impoverished as it was, provided a refuge from the oceanographic catastrophe occurring in the seas beyond it. The oxygen crisis that drove marine anoxia across the late Devonian did not reach the same intensity in freshwater and terrestrial environments, and the organisms that had already committed to survival in those environments were not subject to the same mechanism of extinction that eliminated their marine contemporaries. The silence that followed was not absolute. Life persisted. In the deep ocean, beyond the reach of the most severe anoxic conditions, communities of surviving organisms continued to function. In freshwater systems, fish lineages that had diversified through the Devonian persisted into the Carboniferous, continued to grow, their expansion uninterrupted by the marine catastrophe occurring at their margins, their geochemical influence on the atmosphere and ocean continuing to drive the environmental changes that were themselves part of the system that had pushed the marine world toward collapse. The Devonian extinction did not reset life on Earth. It restructured, eliminating the dominant lineages of one of the most biologically complex periods in marine history, clearing the ecological space into which the survivors would expand and setting the conditions for the biological world that would follow. What was lost was irreplaceable. What remained was enough. After the last pulse of the Devonian extinction, the world was quieter, not silent. Life had not been eliminated, and the processes that sustain biological communities continued operating in the environments where enough had survived to maintain them. But the specific character of Devonian life, the reef systems, the armored predators, the extraordinary diversity of shallow marine communities that had been accumulating complexity for 60 million years, was gone. What remained was a diminished, restructured version of what had existed before, ecologically simplified, geographically contracted, operating at lower diversity across most environments, but alive and capable of the only thing that mattered in the aftermath of a mass extinction: persistence. The fossils left by the Devonian world are distributed across every continent on Earth. They occur in limestone formations that were once shallow tropical seas, now elevated by tectonic forces to positions thousands of meters above sea level. They occur in black shale sequences deposited in anoxic basins, where the absence of oxygen that killed the organisms preserved them. Their soft tissues occasionally retained in forms that normal oxygenated seafloor conditions would have destroyed within days of death. They occur in river sandstones and floodplain mudstones, preserving the remains of freshwater fish and early tetrapods in the sediments of systems that dried, shifted course, and were buried under thousands of meters of subsequent deposition across hundreds of millions of years. The Devonian fossil record is not uniform in its quality or completeness. Like all fossil records, it is a sample, biased toward organisms with mineralized hard parts, toward environments with high rates of sediment deposition, toward geographic regions where subsequent erosion has re-exposed Devonian age rock at the surface. The organisms most likely to be preserved were those with calcified or phosphatic skeletons living in or near environments of active sediment accumulation. Soft-bodied organisms, organisms living in erosional rather than depositional environments, and organisms inhabiting environments where subsequent geological activity has destroyed the rock record are systematically underrepresented or absent. Despite these biases, what has been recovered is extraordinary. The Gogo Formation of Western Australia, deposited in a late Devonian reef environment and subsequently lithified into nodular limestone, has yielded three-dimensionally preserved fish specimens of a quality unmatched in most of the Devonian record. Acid preparation of Gogo nodules dissolves the surrounding carbonate matrix while leaving the phosphatic bone of enclosed fish specimens intact, revealing cranial and postcranial anatomy in 3 dimensions that flat compressed specimens from other localities cannot provide. Gogo material has contributed fundamental information about placoderm anatomy, reproductive biology, including the evidence for viviparity discussed in the context of placoderm evolution, and the internal structure of early vertebrate sensory systems. The specimens recovered from this single locality have revised scientific understanding of Devonian vertebrate biology in ways that decades of work on other assemblages had not achieved. The Miguasha locality in Quebec, a UNESCO World Heritage Site, preserves a late Devonian estuarine community of exceptional diversity and completeness. Fish specimens from Miguasha include some of the best preserved examples of lobe-finned fish anatomy in the fossil record, with material that has been central to understanding the sequence of anatomical changes involved in the water-to-land transition. A low-energy estuary with periodic, anoxic bottom conditions that inhibited scavenger activity and promoted preservation produced a fossil assemblage whose biological information content has sustained productive scientific research for over a century and continues to yield new findings as preparation and imaging techniques improve. The Rhynie Chert of Scotland, though predating the main Devonian interval discussed in this account, preserves an early Devonian terrestrial and freshwater community in silicified detail of unparalleled resolution. Cellular level preservation of plant tissue, fungal structures, and early arthropods in Rhynie material has provided information about the biology of the earliest terrestrial ecosystems that no other locality approaches, documenting the mycorrhizal associations between fungi and plant roots, the structure of early vascular plant tissue, and the anatomy of the arthropods that inhabited the margins of early Devonian hot spring environments. The legacy of the Devonian extends beyond the fossils themselves. The first forests of the Devonian permanently altered the carbon cycle of this planet. The drawdown of atmospheric driven by forest expansion and the associated acceleration of silicate weathering initiated a long-term trend toward lower atmospheric carbon dioxide concentrations that, with fluctuations, has persisted across subsequent geological time. The oxygen produced by Devonian and Carboniferous forests accumulated in the atmosphere, eventually reaching concentrations that made the Carboniferous one of the highest oxygen intervals in Earth's history. With consequences for the size of insects, the frequency of wildfires, and the physiology of the early terrestrial vertebrates evolving within those oxygen-rich environments. The tetrapods that survived the end Devonian extinction carried with them the anatomical foundations of every subsequent vertebrate lineage on land, the limb skeleton of all terrestrial vertebrates, the humerus, radius, and ulna of the forelimb, the femur, tibia, and fibula of the hind limb, was established in its fundamental configuration in the Devonian, in animals whose primary environment was still shallow water, the digits, the neck, the ear structures capable of detecting airborne sound, the lungs. All of these features, present in every terrestrial vertebrate alive today, trace their origins to evolutionary changes that occurred in Devonian fish and early tetrapods navigating the margins of Devonian water bodies. Every mammal, every bird, every reptile, every amphibian alive today is a descendant of organisms that survived the end Devonian extinction and carried those anatomical innovations forward into subsequent geological time. The reef gap that followed the Devonian extinction lasted approximately 10 to 15 million years. When reef systems recovered in the Carboniferous, they were built by different organisms. ghost corals persisted in reduced form, and new reef-building communities assembled from the survivors of the extinction and the lineages that diversified in its aftermath. The ecological architecture of these post-Devonian reefs was different from the stromatoporoid-dominated systems of the Middle Devonian, and the communities they supported reflected the changed composition of post-extinction marine fauna. The sharks that diversified into the ecological space vacated by placoderms became, across the Carboniferous and subsequent periods, the dominant large predators of marine environments in ways they had not been during the Devonian, the lineages that would eventually produce the diversity of modern chondrichthyans, including the forms that represent the apex marine predators of the contemporary ocean, trace their expanded ecological dominance to the competitive restructuring that followed placoderm extinction. The Devonian ended. The world it had built, the reef systems, the armored fish, the first forests, the ancient shallow seas, passed into the geological record, are compressed under subsequent deposition, transformed by heat and pressure, and time into the rock that human researchers have been extracting, describing, and interpreting for 2 centuries. What those rocks contain is a record of a world that existed for 60 million years, and was then fundamentally altered by a combination of environmental changes, whose causes were, in part, biological, produced by the same living systems whose destruction they ultimately contributed to. The Devonian world did not simply end. It left the conditions for everything that followed. In the structure of vertebrate limbs, in the chemistry of the atmosphere, in the carbon stored in ancient rock, in the empty ecological space that survivors moved into, and from which the biological complexity of subsequent geological periods slowly, unevenly, and without direction rebuilt itself. The stone remembers, and in the stone, the Devonian persists. Compressed, mineralized, and silent, but present, in every fossil that carries the record of a world that was already ancient when it disappeared.